Hello everyone, myself Mr. Namit Kudatarkar. So today I am going to talk about serotonergic neurotransmission and its receptors. Learning objectives. By the end of the topic, learners will be able to know synthesis, storage and release of serotonergic neurotransmitters and characteristics of serotonergic neurotransmitters. So before we move towards the main content, just know the locations and what are the functions of these serotonergic neurotransmitters. So serotonergic projections to the spinal cord are mainly involved in the pain perception, visceral regulation and motor control, and serotonergic projections to the forebrain are involved in modulating mood, cognition and neuroendocrine function. So the primary mode of 5-HT is varicocytes, and Raphe nuclei has these 5-HT containing cells which project throughout the cerebral cortex. So now we will see how these serotonergic neurotransmitters are being synthesized, regulated, stored and released. So in this diagram we can see the one which is present above is this presynaptic neuron, the one which is present below is the postsynaptic neuron, and the gap between the presynaptic neuron and the postsynaptic neuron is called as a synaptic cleft. Now in the presynaptic neuron, sodium along with an amino acid which is called as tryptophan will enter this presynaptic neuron. So here there is a transporter which we call it as a aromatic L-amino acid transporter. So this aromatic L-amino acid transporter will take up the amino acid, that is in this case it is what? Tryptophan, right? So once tryptophan has entered the presynaptic neuron, it will get converted to 5-hydroxytryptophan in the presence of an enzyme called as tryptophan hydroxylase. So this 5-hydroxytryptophan, again in the presence of an enzyme, that is aromatic amino acid decarboxylase, will get converted to 5-hydroxytryptamine, that is 5-HT. Now this is how the 5-HT is been synthesized. The next one after synthesis will be the storage. So 5-hydroxytryptamine needs to be stored somewhere, right? So where it is going to be stored? It is going to be stored in the vesicles. So what is this vesicle? It is a storage compartment. Okay. So this 5-HT which is being synthesized is being stored here. So how it is taken up within this vesicle? So there is a transporter here. This pink portion which you can see here, it is a transporter which is called as vesicular monoamine transporter. It is also abbreviated as VMAT. Right? So what this VMAT will do? It will take up this synthesized 5-hydroxytryptamine and it will take into the vesicle for storage. So this was all about the storage. Now we will see how it is being released. Right? So whenever there is a nerve impulse which is generated, it will cause an action potential, which in turn there is a calcium channel here, right, in this portion. So the extracellular component, the calcium which is present in the extracellular component will enter the intracellular component of the cell through this calcium channel which is present at this corner. Okay, so it is mainly involved in intake of the calcium. So when there is an excess amount of calcium, right, within this presynaptic neuron, it will lead to a depolarization, and whatever the 5-HT is present in this vesicle, okay, it will be thrown out by means of exocytosis, that is from the presynaptic neuron to the synaptic cleft, right? So this is how it is been released. 5-HT is released. Now here there are three criteria. So the first criteria will be the 5-HT neurotransmitter which is being released, it will go and bind to the postsynaptic receptors. So these are the receptors which are present on the postsynaptic neuron. It is called as a postsynaptic receptors. So here there are different types of postsynaptic receptors. So 5-HT1A, 2A 2CS, 5HT4, 5HT3. There are a number of 5HT receptors which we will be seeing it in the upcoming slide. So first criteria is it will go and bind to the postsynaptic receptors, and it will cause the desired effect by going through the particular pathway like IP3DAG, or by going through ion channel or CAMP. Right, so based upon the different pathways, there will be different actions which will be formed or produced. Okay, the second criteria will be this 5HT which is present in the synaptic cleft. It will also go and bind to the 5HT1D autoreceptors which is present in the presynaptic neuron. Right, so it will provide a feedback inhibition. Okay, the third criteria will be this 5HT, it will be taken up back into the presynaptic vesicles, okay, or it will be degraded with the help of an enzyme, that is monoamine oxidase enzyme. So how it is taken back by the presynaptic neuron? By the plasma membrane serotonin transporter, SERT. Right, so it is taken back by the vesicle, and it is degraded with the help of an enzyme, that is mitochondrial enzyme, monoamine oxidase enzyme. So again, in monoamine oxidase enzyme there are two isoforms, that is one will be monoamine oxidase A and monoamine oxidase B. So in this case it is monoamine oxidase A enzyme. Right, so this is all about the synthesis, regulation, storage and release of 5HT neurotransmitters. So now we will see the characteristics of serotonergic receptors. So these serotonergic receptors are mainly of different types. So one you can see here, there are again subtypes like 5HT1, there is 2, 3 and 4, 5 and 6, right? So in one again there are subtypes like 5HT1A, 1B, 1D, 1E and 1F. In 2 it is 5HT2A, 2B and 2C. Then there is 5HT3, 5HT4, 5HT5A and 5HT5B and 5HT6. So here you can see the signaling mechanisms. 5HT1A, B, D, E, F, right? All undergoes, they act on the G protein. These are G protein receptors and they act through GI, right? So that will lead to what? Decreased CAMP, that is cyclic adenosine monophosphate. Okay, so what will be the distribution of this 5HT1A? It is mainly present in the raphe nuclei and hippocampus, and the activation of this will lead to, it will regulate sleep, feeding and anxiety. Next one is the 5HT1B. So it is mainly found in the substantia nigra. The next receptor is 5HT1D, which you find it in the brain, and its main effect is vasoconstriction. 5HT1E is mainly found in cortex and hippocampus, and it's mainly involved in memory. 5HT1F is found in globus pallidus and putamen, and it's involved in anxiety and vasoconstriction. 5HT2A find it in platelets and cerebral cortex, and the effects are mainly cellular excitation and muscle contraction. 5HT2B you find it in the stomach, and it's mainly involved in maintaining the appetite. 5HT2C you find it in hippocampus and substantia nigra, and it is also involved in anxiety. 5HT3 is mainly found in arena postrema and enteric nerves, and is mainly involved in vomiting. 5HT4 is found in cortex and smooth muscle, and its effects is in gut motility. 5HT5A and B you find it in brain, along with 5HT56 also you find it in brain. So the function of 5HT A and B will be in locomotion and sleep, and 5HT6 will be in cognition and learning. So these are the some of the characteristics of the serotonergic receptors. So it is a reference. Thank you.